Yo, so today we're going to do embryology again, attempt 2. Because there's no presentation slides, I decided to just make notes like normal notes for the videos, asking questions and having the answer right below it so then I can just walk through it. Might take a bit longer because I know everything now, but yeah, I think I'll do the same thing with diversity of life, which I'll start in 2 days. I'll have to do epigenetics because I didn't learn that. So anyways, so difference between ovum and an oocyte. So ovum is when it's in the ovum is like after what's that thing called, ovulation, and then oocyte is within the ovary. Yeah, so what are different layers of the ovum? So the most superficial outer layer will be the corona radiata, so this is like the outermost. This is like, because it's the outermost, it'll be the protective layer, and it'll be made up of granulosa / follicular cells. So this is like kind of what we were talking about, like the follicle being developed in the menstrual cycle alongside the egg. And then what it'll do is it'll help care for the egg after ovulation. So corona radiata, care for eggs after ovulation, made up of granulosa and follicular cells. So now going past the corona radiata, we're gonna have the zona pellucida / the vitelline membrane. This is where sperm binding proteins are. This is like the main important thing, and this is like a jelly-like coating with glycoproteins. So these sperm binding proteins, those are responsible for preventing, like, polyspermy, or like making sure the right species fertilizes the right species. So yeah. Then we got the perivitelline space. So this is just an empty space, and this is mainly used for preventing polyspermy, which I'll get into later. So just to go over, we got 3 layers to the ovum. We got the corona radiata, the zona pellucida, and the intervillum space, or no, the perivitelline space. I'm trying to close my eyes and say this. So 3 layers of the embryo, or of the ovum. We're gonna have the corona radiata, the outermost part, which is made up of glycoproteins. Provides like protective layer made up of granulosa and follicular cells, care for eggs and ovulation. Then after that we got the zona pellucida, and then we got the we just got the perivitelline space. Corona radiata, zona pellucida, perivitelline space. Now within the plasma membrane, this is what's going to be after the perivitelline space. So the perivitelline space separates the zona pellucida and the plasma membrane of the egg. So within the plasma membrane, we're going to have the organelles, nucleus, containing the haplogenetic information, the mitochondria, which is the mom's mitochondria, which will be in the fetus or the offspring. We got the endoplasmic reticulum, which stores this brightness is cool. This will store like calcium or sodium ions. Then the cortical granules, those are kind of what bind to the plasma membrane to prevent polyspermy, which we'll get into later, the cortical reaction. Okay, now moving on to the anatomy of the sperm, we have the head of the sperm. This will contain the haploid nucleus plus the acrosomes, which are responsible for breaking down the corona radiata. Or not even the corona radiata, it's used for the vitelline membrane. Got the mid piece, which is the mitochondria to generate energy, and then the tail is the axial filament, the flagellum, that just flip-flops around to propel the sperm. Okay, so now moving on to the process of capacitation. So when will it occur? It occurs when we enter the female reproductive tract. Who is we? I mean, it's like freaking the sperm. Okay, wait, that's not even funny. Why am I laughing, bro? It's too late. So the function of the glycoprotein coat on sperm is like a protective layer. The example they give is like a mom giving their child a coat before going into a snowstorm. So these glycoproteins will prevent the sperm from dying in the reproductive trait of the female. And then also, that's basically all. It's just like a protective coating. It doesn't really do too much. Just protecting it, kind of immobilizing it, and then that's what capacitation will allow. So it'll be added in the epididymis of the testes, There's the, okay, you kind of forgot that there's like the seminiferous tubules, the Van Leydig cells, the Sertoli cells. Don't really remember what the epididymis did. It's all the advanced deferens. Yo, okay, what is the epididymis? Where is my mouse? Oh, the epididymis is kind of connecting the. Oh, that's where they mature finally. Like that's the final storage site. So testes will. Okay, that makes sense. So it's like the middle. So what's the acronym called then? I don't remember. Okay, wait. Now I want to know. 7 Up. Okay, so what does capacitation do? It does 3 things. So the digestive enzymes from the uterus will break down that glycoprotein coat of the sperm. Then destabilization will also occur. The membrane will be released from the acrosome, kind of like from the top of the head, to make the acrosome be able to release faster. And hyperactivation, you'll start moving fast. Cause unknown. Maybe the glycoprotein coat was like immobilizing it. So what is the zygote? The zygote is simply just a fertilized egg, like a diploid cell, sperm and egg fusion. So what's the process to fertilization? So first, the sperm will go into the reproductive tract, then go into uterus, and it'll start flowing towards the fallopian tubes, the oviduct. So now the first step, cornea radiata, like the first superficial layer, the sperm will travel to the oviduct and contact the egg, and it'll go through this cornea radiata. Now it'll reach the zona pellucida. The zona pellucida, keep in mind, that's coated with the glycoproteins that have the sperm cell receptors. So once the sperm reaches that sperm cell, like that CP3 or whatever, if it matches, then the acrosome reaction will occur, and the acrosin, which is within the acrosome, like acrosin is like the actual like digestive enzyme, like the hydrolytic enzyme, and it'll break down that layer and it'll go into the perivitelin space. So this space is very tight, so it'll be like the side of the sperm that will bind to the plasma membrane and then release its contents into the plasma, like just into the egg cell, and then fuse with the nucleus, and then boom, touchdown. So fertilization will trigger 2 things here. So first, the egg will no longer be arrested in metaphase 2. If you recall to the cell division or reproductive system, these egg cells are arrested or stopped at metaphase 2 after puberty. So we'll be able to finish metaphase 2, finish meiosis 2, and then a polar body will be created from this division of meiosis 2. Now, cleavage will begin from here, and yeah, like it's just the first type of division that occurs. So that's something to know. Like, what is the first type of division that a fertilized, that a zygote will go through? It will not be like mitosis, it'll be like cleavage. So what is polyspermy? That's just multiple sperm fusing to the ovum. It's not good because you got too much DNA. So how is this prevented? So there's 2 ways. So there's the fast block. So first, what will happen is that any plus channels along the egg plasma membrane, they'll open up and it'll depolarize the membrane. Similar to a neuron action potential, will go from negative to positive. So the egg is currently negative, but the Na plus channels going in, that'll make the cell positive, and then that'll prevent other sperm from entering. Now there's a slow block as well. When depolarization will occur, the calcium ions will be released from the endoplasmic reticulum, and that'll cause the cortical reaction, as these cortical granules, which we talked about earlier, they will fuse to the plasma membrane and then put the content, the calcium 2 plus, into this empty space, the perivitelin space, and then that'll cause it to kind of swell up and block the ZP receptors, also cause detachment from the plasma membrane. So yeah. Okay, so cleavage, what is unchanged? So cell mass is unchanged like cytoplasm mass. Cleavage is just like cell dividing into smaller pieces, but you're not really like changing the mass. So blastomeres, these are the results of cleavage. Difference between meroblastic and holoblastic. So meroblastic is unequal division, so partial division due to yolk. You're going to get unequal cell size because of this partial division. Holoblastic, that's equal division. Holoblastic, I want to say, is usually faster because you're not going through the yolk, while meroblastic will take longer. But I feel like that's related to like the vegetal and animal pole, which we'll get to later. So when will you see a still is a determined blastomere or differentiated blastomere? So a determined blastomere, it knows what it wants to be, but it hasn't undergone differentiation. A differentiated blastomere, that's simply just a blastomere that's changed and is like ready to become what it wants to be. So difference in determinate and indeterminate cleavage, what happens when you isolate them? So the determinate cleavage, you're going to get a cell that divides into two blastomeres. They know what they're going to do, and they're determined to be there. So you're going to get a specific tissue type if you isolate it. Indeterminate cleavage is when you have two blastomeres, but they don't know what they want to be. So you could take any of these cells, isolate it, and it'll develop into the full organism because they haven't differentiated yet because they're not determined. You have to be, in order to be determined, you have to have done differentiation. In order to do differentiation, you need to be determined. So difference in animal pole and vegetal pole. So animal pole will be no yolk, vegetal pole has a yolk. So the top will divide faster than the bottom. The vegetal, maybe it doesn't have to be top or bottom, but the vegetal pole will divide slow. Radial cleavage, this is like balanced cleavage. This is like proper, equal, parallel, perpendicular to polar axis. So you're going to divide, like in orientation, spiral cleavage, there's no alignment, it's kind of a mess. You're going to have unequal division and an unorganized organism. So macromeres and micromeres, those are the resulting cells from this disorganized division. So macromeres will be the bigger portions, micromeres will be the smaller portions. Protostomes, they will undergo determinate and spiral cleavage. So protostomes, those are when the GI tract or the blastopore will develop into the mouth first, while deuterostomes, they'll develop into the other end. But protostomes will undergo determinate and spiral cleavage, while deuterostomes will undergo indeterminate and radial cleavage. That means humans must be deuterostomes, right? Yeah, I don't know. Okay, so now we're going to talk about we're just continuing on with the progression of the zygote development. Okay, so first we have fertilization, zygotes formed, cleavage undergoes, blastulation, gastrulation, organogenesis, and neurulation. So currently we're going to talk about cleavage, blastulation, etc. So the zygote will undergo cleavage and get to a blastomere, which is simply just like a 32-cell zygote, which is also happens to be called a morula. Nice. So this cleavage is occurring as it moves through the oviduct, and it's contained within the zona pellucida. Why do we need the zona pellucida? Well, it's important for preventing improper implantation. It prevents the morula from attaching too early, and we'll talk about that a bit later in the gastrulation of the importance of the trophoblast and all that. But everything sounds the same, bro. It's so dumb. What? It's probably one of my diversity of life is my least favorite. It's gotta be close second. Not close, it's kind of far second, but I enjoy everything else so much more than this, bro. Sure, it's cool to know, but I don't want to be talking about blastula, morula, mastolis, all this BS. So yeah, anyways, cleavage occurs or contained within the zona pellucida. Yeah, it helps with immune protection as well because, the mom has this random thing inside of her. So it protects the immune system from like trying to attack it. So blastulation will occur when the morula turns into a blastula, which is called a blastocyst in humans. What's the purpose of that, bruh? Blastocyst in mammals, I should say. So how's the blastula different from the morula? The blastula, that has a hollow cavity which contains the blastocele. So blastulation is simply just the forming of the blastocele. Gastrulation is when we start to see an infold on the blastula. So we're going to fold into the blastocele, and then we're going to fold that blastopore, that inward fold opening kind of area. That's what the blastopore is, and that'll determine if you're a protostome or a deuterostome, determining if you develop into the mouth or the anus. The archenteron is, if you continue to fold inwards from the blastopore, you're going to end up kind of dividing the cell, right? So now the archenteron is that space between the two kind of half cells that you made, and this will develop into the like GI tract, like the digestive tract. This is also when germ layers are introduced. We're going to have 3 cell types: the ectoderm, the mesoderm, and the endoderm, I believe. So now moving on, we're going to talk about mammals, or like the base model of gastrulation. So we're at the blastocyst stage. We have the blastocele, and so at this step, the cells will differentiate into the trophoblast and the inner cell mass. So keep in mind we're at the blastula stage, so this is when we have that kind of space in the middle. Now we're going to differentiate the cells. See, that's what I don't really get. Is like, I guess gastrulation is always going to be the primitive cell differentiation, but like, bro, I don't know. So the blastocyst stage, we're going to get the trophoblast and the inner cell mass. So embryoblast is the inner part, trophoblast is the outer part. That's why I was talking trophoblast prevention in implantation. But anyways, the trophoblast is the exterior, and that'll form the placenta, and that's also what's responsible for implantation. That's why I need the zona pellucida, because the trophoblast is the outermost layer. So if we don't have the zona pellucida, the trophoblast will attach the uterine wall and implant, and we don't want that to happen too early. So that's what zona hatching is. For implantation to occur, the zona pellucida must be broken out of to expose the trophoblast, and that is the important fact about zona pellucida. So also the trophoblast will form the placenta, which is responsible for human cortico something, HCG, but that's just used to like maintain the uterine walls rather than the corpus luteum having to make progesterone and all that. Or no, it'll maintain, I think that, so the placenta is going to make HCG, and that HCG is going to maintain the uterine wall and also the corpus luteum, which is reproductive, but I don't think that's important. So zona hatching, we already talked about that. What is the inner cell mass comprised of? It's going to comprise of the epiblast and the hypoblast. So we're going to think of these as a bilaminar disc. So it's going to be a disc with the epiblast on the top and the hypoblast on the bottom. Now, with gastrulation occurring, 3 layers are present. So how does this happen? So very first, the epiblast layer will thicken, and it will form something called the periodic streak, which is kind of an elongated blastopore in mammals. And you're going to start forming 2 bumps with kind of a groove in the middle, and that's going to be called the periodic groove. Now some cells, which are called mesenchyme cells, which are of the epiblast layer, so the top part of the layer, they will lose adhesion and they will migrate towards the groove and form the inner mesoderm layer. We're going to go from a bilaminar disc to a trilaminar disc. Hooray. So now talking about frogs. I don't really know why we need to know this, but I guess we do. So they're an exception. They have a large yolk sac, but they will go through holoblastic rather than meroblastic. So you're going to get unequal distribution of yolk rather than unequal distribution of cells or unequal division. Sperm will fuse at the animal pole, which is the side with no yolk, and then the gray crescent will form on the opposite side. Why? Because the cell will rotate 30 degrees, and that'll cause a gray area where the cytoplasm is breached into from the yolk sac. This will be at the opposite of the sperm. This will be opposite of the sperm. And this cleavage will occur in a way that the orientation of all these cells is proper. So gastrulation occurs here when the gray cells begin to fold in to form the dorsal lip, and that's like the blastopore. There will be no periodic streak because that's a mammal thing. The archenteron will fill up the space where the blastocele once was, and then you're going to get a yolk plug which kind of seals the two. So yeah, I'm not really too sure about that. So now, bro, what? Okay, so now we're talking about sea urchins. This is the primary model of division, not too complicated. The only difference is that instead of getting an elongated blastopore, or whatever that thing is called, the primitive groove elongated blastopore, we're going to have a circular blastopore. Now talking about egg-laying animals, they undergo meroblastic because they have the yolk. In fact, they have a lot of yolk that the embryo is squished, and it's actually called a blastodisc, not a blastula. So what is a blastula? A blastula is just what it's called when it's like a result. Wait, what? Blastomere? What's the difference between blastomere and blastula, bro? Come on, bro. Don't do this to me, bro. Blastulation occurs so the blastula is just with the hollow cavity. So the blastula will be called the blastoplate or something because of the yolk. The chalaza. The chalaza are responsible for stabilizing the yolk. The albumin is the egg white, and common between them two, they have the privative streak with the elongated blastopore as compared to the sea urchin. Moving on to anamniotes. Amniotes, they have the amnion sac, while anamniotes do not. So birds, reptiles, and mammals, they have the amniote. Amphibians and fish do not. So what is the amniote, you may ask? The amnion is just shock absorption. So we're going to compare amniotes between egg-laying and placental. So the chorion, so there's four main things. There's the chorion, the allantois, the yolk sac, and the amnion. So like I said, amnion is used for shock absorption. Yolk sac is just nutrients basically, but for placental, you get your nutrients from the freaking placenta. So you're going to do red blood cell formation in the placenta. The allantois is mainly for waste. So for eggs, it'll store waste. Placental, it'll transport waste to the placenta, which will turn into the umbilical cord. The chorion, that is for kind of gas exchange. So eggs, again, that'll just do gas exchange, while the placental, that'll form the placenta. Okay, so now the nodal, oh my god, bro. Okay, so this is kind of like the complicated part. We're in like the organogenesis neurulation area. So the nodal cord is derived from the mesoderm layer. So what is induction? It's kind of when you influence, like one cell will influence other cells to develop, and this is done by the nodal cord to develop the other things like the neural tube. So the neural tube is developed. How does it happen? So the nodal cord will stimulate the ectoderm to thicken and form a neural plate. So keep in mind the nodal cord is the mesoderm. So above that we're going to have the ectoderm, and this ectoderm is going to thicken. the neural plate will form, which is the thickened portion of it. Then there will be an inward fold and cause neural folds and a neural groove. Now this neural groove is the space in between the two folds. It's like a canyon. Now this neural folds will connect, and then we're gonna, hopefully you can visualize it, we're gonna form a tube because two folds folded over a groove, boom, neural tube. That will turn into the CNS. This is what makes a neurula. A neurula is simply just a developing embryo that has undergone neurulation. So neural crest cells are formed from this as well. There's a process called delamination. As the neural folds, the ectoderm, are formed, these neural crest cells will break off through delamination. So the ectoderm, the outermost layer, that'll form the outer part. So epidermis, like skin, hair, nails, keratin maybe. The neural tube is also from the ectoderm, the adrenal medulla, and the neural crest, so the skull. Now talking about the endoderm, the inner part, that's going to take your epithelial lining, your tracts, like your urinary tract, your digestive tract, liver, lungs, pancreas, the organs that are used for those, thyroid and thymus, those control metabolism, so it's kind of like related. Now the mesoderm is everything in between, so that's bones, cartilage, connective tissue, circulatory system, the adrenal cortex, and the notochord cord. Notochord cord was the mesoderm. So yeah. There's also neural crest cells. Okay, that's all.